the complete the afternoon, we are going to have Dr. Anne-Marie Fortuna come and visit with us about microbiology and what the microbiology are doing in the compost and how they're changing things, making things different, transferring nitrogen from one form to another, and anything else. She has some demonstration stuff, and we have a few things to look at over. And Anne-Marie came from Washington, I believe, Washington State.
Okay, so I've been working with compost for a long time across a range of states, Michigan, Washington State, and now here in North Dakota. And largely I look at the nutrient management aspects similar to Chris. I decided to start out by saying that most people think of the microbiology as a black box. I mean, they know that there are microorganisms in the soil, in the compost piles, but we really don't pay that much attention to them. And if we manage our system well, we can, to a certain degree, ignore them. But that doesn't mean that they're not important and that they're not influencing the nutrient cycling and other things that we're trying to do. So I dubbed this the Fabulous Five, kind of like the Fab Four. These 5 organisms here represent the majority of the organisms in the compost pile, and the ones in the center are the ones that we find in most numbers. So actinomycetes is a bacteria, but it's kind of interesting. They look a lot like fungi, and they have enzymes a lot like fungi. And I think Chris mentioned enzymes. So enzymes are important because they do 2 things. They break down the organic carbon so that the microbe can use that carbon, and then in turn, the carbon that remains that they don't use for liquid food is also transformed and becomes more recalcitrant, and that's why the pile, starts out tall and then eventually, it compresses because they've used a lot of that material and they've made it smaller. So actinomycetes are really important because they have those enzyme complexes, and fungi are the ones that have the most what we call extracellular enzymes that come out of their cell. Like if we can take something out of our body and break down these larger, more complicated carbons, like lignin in plant tissue. Wood has a lot of lignin in it. And then bacteria, and you can see here there's 100 million to 1 billion of them, so they make up quite a bit of the biomass there. They also are able to decompose things more quickly, but they are not able to get to the more recalcitrant stuff. And then the 2 guys on the bottom are sort of backup players, really. Protozoans are also in the compost pile and in the soil and water films that adhere to organic material. And then they also use the carbon. And then rotifers, they're actually kind of similar to a diatom, or they're multicellular there actually, and they also degrade carbon. When these organisms take in carbon, they generate heat. And so how the compost pile heats up is that they're using part of the energy, making their own making their cells doubling, but they're not very efficient. So if it's aerobic, we have a more efficient system. So if your pile goes anaerobic, the bacterial efficiency drops too, and so things don't work well. And so that's essentially why you need to aerate that pile and keep a certain temperature, moisture scenario inside it. And so as they are heating up and using that material, they can actually kind of get to the point where they kill themselves off if we're not careful, and introduce more oxygen, introduce some moisture. So what I was hoping to communicate to you guys are 3 things. What are the conditions that we want to have in a compost pile? Anyway, we have to look at chemical, physical, and biological conditions, and I'll talk about how we can look at the pile and know that certain conditions exist. Then we're going to talk about the phases of the compost pile. So, thermophilic when it heats way up, mesophilic when it goes back closer to room temperature, and then the curing phase where there's really no heat inside it. And then we'll relate each of those phases to groups of organisms. So basically by the end of the talk, you'll understand how all these properties like the porosity and things like that relate to microbial communities and what that means when you're trying to judge whether your compost pile is done or not. And then I'm going to give you some quick tests, some quick ways for you to monitor your pile. And you won't have to try to extract any microorganisms, but you'll know what they're doing. based on some quick tests. So this picture is residue, actually, and it's a bunch of fungi and bacteria decomposing something that looks maybe like a corn residue. So you can see there's just like a huge consortium, a group of organisms working together to reduce that material into something that they can use. There's the 3 stages: the thermophilic, mesophilic, and then curing stage. And this is probably towards the beginning of the stage. So here is a diagram with temperature on the side, and I apologize that it's in Celsius. But I believe like 37 Celsius is about 100 Fahrenheit. This range down here is room temperature, just above 20. And so you can see at the top, at the thermophilic phase, it says high rate of decomposition and hygienization. It's a new word for me. So that's where you burn off the weak seeds, you kill them, and in theory, you should have your pathogens under control. So the heating phase is important for a number of reasons, not just for getting your carbon into a more stable state in your nutrients in a more stable state, but it's also very sanitizing, and especially with the animal carcass. I guess you probably talked a little bit about that. Sorry, to keep using. That's why we can get away with doing that. And in theory, there are enough enzymes and high heat in a compost pile that they think that prions, like mad cow prions, could even be reduced to significant low numbers such that they get the state. Obviously, the microorganisms probably don't look like this, but keep in mind I use this for class too and for outreach. So, you know, unlike humans, bacteria have a wide range of temperatures that they can survive in. If we get to about 100 Fahrenheit for, you know, a constant period, we're going to have a hard time regulating our body heat. And so, because we have different cells, you know, and inside that are not stable at higher or lower temperatures, we can sit in an ice pack, for example, like the psychophiles organism there, which we're not going to talk about because he wouldn't be in a compost pile. We have mesophiles. We're mesophiles, really. We operate, you know, in a nice range, a comfortable range of about 40 to, you know, 100 Fahrenheit. So the inside of your refrigerator is about 45 Fahrenheit. So, you know, when you want to put on a coat, that's about the end of the mesophilic range. And then when you can't sit outside very long without getting heat exhaustion, that's probably the end of the mesophilic phase. So in the pile, there are thermophilic and mesophilic organisms. And you're probably thinking, well, how is that possible, right? Well, on the outside of the pile, it's a lot cooler than the inside. So in the beginning, you're starting out with mesophilic organisms, the compost pile is heating up, your population shifts, you kill off most of your mesophiles during the current phase, and then as it cools back down again on the outside of the pile, these organisms come in. So that's why we often say, you know, that microbes are a black box, because we never have to add them, right? I mean, we add rhizobia to our soybeans, but for the most part, any of our soil organisms are just there. And we're creating conditions in which certain organisms are favored, and they do the work for us. You know, our corn plants have mycorrhizae, and those mycorrhizae are fungi, and they're in the soil. Without them, even the fertilizer you put down aren't going to be available to them. So the microbes are really important. So again, properties of the compost pile and the waste that we're adding, I think you guys covered pretty well. But I want to talk a little bit about that. We need to have what I'm calling substrate. That's the food for the organisms. That's our, you know, what we're putting in there, residues, wood chips, the things that we're going to compost, manure. And those properties have, those things have chemical properties. So Chris talked about the salt concentration, the SAR, calcium, magnesium, and sodium in the pile, and how that gets really high over time. Those salts could, you know, have a negative net impact on the field. So chemical properties in the pile are important too, because these organisms need to have certain elements to utilize. And if the pressure, the osmotic pressure from salts is too high, they don't function very well either. So we don't want to add, for example, a waste that's high salt, like a lot of food waste can have high sodium. So So if we wanted to add it to a pile, we'd have to think about that. So that's where the chemical properties come in. The physical properties are more about aeration and water content. So if you think about these peanut chips, you know, there's like lots of air between them, and they're very fluffy. So something like that in nature would be maybe these little pine cone and bark chips. So, you know, a lot of times we put wood chips and bark chips, bigger things, into piles, and they maybe don't have a lot of nutrient value, but when we put that in with something like the mortality pile or the manure, then we balance our nutrients with our pore space. And so we create the right bulk density so that we get enough air and water in there for things to cook. So it's really, you know, getting the right balance. And I'm just putting this up here not because you should know all these details, but if you look at, for example, the pine needles and wood, like I just showed you guys, they're in the range of 400:1 to 200:1 for their C to N ratio. Now, a C to N ratio that would allow you to get a high enough rate to compost would be more like 35:1. 35 units of carbon to 1 unit of nitrogen. So if we just put pine needles and wood chips in, we wouldn't get any heat in our compost pile. On the other end, the animal carcass is 5:1. So you can see that there's ways of mixing these things together that allows us to make that right chemical environment. And the bacteria and the fungi themselves, they have a carbon to nitrogen ratio, and so do we. So the bacteria are cranking in the compost pile, and the fungi too. They have a different C-N ratio, and they're using what they need, not perfectly efficiently. We talked about that. And what they excrete then becomes available to other things. And so after they run out of carbon or nitrogen, and they stall. So that's why we need to keep monitoring our pile. So as I believe I mentioned in the previous slide, you want to get a balance of about 25 to 35 for the carbon to nitrogen range. And if you go more than about 40 on your C to N ratio, 40 units of carbon to nitrogen, you get a mobilization. You know, many materials start out with a certain set of chemical properties, and as they get composted, they change. You know, so you run out of carbon, but things like pH, for example, some compounds start out acid, and then they become more basic or neutral. And I guess you can equate that to fertilizer applications. If you apply urea, you convert urea into ammonium, and that gets converted into nitrate, and hydrogens are produced during that process by bacteria. And that does the opposite, it decreases the pH. So in the compost pile, you often start out with acid, and then through the reactions that occur during the microbial breakdown, you get basic conditions. So that's a benefit in most places. In North Dakota, we do have a lot of soils that have high calcium carbonates, so that, you know, probably wouldn't be a drop in the bucket, but wouldn't necessarily be as beneficial.
And when you talk about the carbon to nitrogen ratio, does it matter to you in which form that carbon is locked up? So lignin versus cellulose versus...
Right. Yes, lignin is a carbon compound, and one carbon compound is not equivalent to another, because some carbons are bound side by side in a single chain. So if you're thinking about, let's say, a rope, if you have, you know, one thread in a rope and you need to cut it, it's going to be way easier than if you have something that have 3 or 4 cords wrapped around each other. So, and then you can take that analogy further and you can say, you know, some carbons don't look like a chain. In fact, they are bound not just by single chemical bonds, but double bonds, and they are not necessarily straight. They can, you know, have different conformations, and it's hard for organisms to break those particular bonds. It's harder. It's like different metals have different properties. You know, some are softer, some are stronger. So chemical bonds are like that too. And so the lignin in the wood is this really complicated structure. It doesn't even have a symmetry per se. It's sort of amorphous, meaning it doesn't have an exact shape. So it's, if you had nitrogen bound to that, it be like in a cage. It would be like the claw or top of the nutrient, and so it would not be readily available. So when we compost, we have the tendency, the organisms tend to select for the straight chain carbons, and they leave behind the things we call humic, which are these amorphous, complicated carbon structures that bind nutrients that are harder for the organisms to get to and to break down to utilize the nutrients in. So that's part of what we want to do in composting. We want to stabilize our nutrients, and we want something that's going to persist, that's going to provide not just nutrients, but it will also, improve tilth, etc. So the composting process itself, as Chris also pointed out, microbes also build carbon as they excrete things, and those carbon compounds are also typically not very readily available. They're fairly complex. Yes, so different forms of carbon are different, and when you're making your compost pile, people usually, like with the microbes, ignore that aspect of it, but it isn't a critical piece, and it's called soil quality, not soil quality, residue quality, for example, kind of hits that point. It's what's your C/N ratio, and then how is that carbon and nitrogen bound. How would you figure out what your bulk density was in a not very complicated way? If you have something like this container here, and you knew its volume, and you could weigh the amount of compost that you put in here, you could calculate your bulk density, and that would give you an idea of what the pore space is. What they've done is, so that farmers and other lay people can figure these things out fairly easily, they say, fill this up to this line here, and if you have this jar and you put 30 grams of compost in it, then that would be equivalent to 500 pounds, yard inches, or cubic yards, pounds per cubic yard. So anyway, if you carefully sampled this, and this represented a larger pile, you can figure out your density by just filling this up to the line and weighing it. So, but basically, you need to be able to weigh your compost, and you need to put it into a known volume, and from that, you can calculate your density. If you didn't know what the density of this jar was, you could put something of a known weight, like water, which is 1 gram per centimeter cubed, and you would be able to know your volume. So it's just a volume by weight. So back to the other physical properties. So how much water to air do we want? We want about 40 to 60% water to air, like pore space. Anyway, so you're going to shoot for about 40 to 60%. Okay, so this is our compost pile. And it essentially has the inputs, the raw materials. So those are organic materials that have carbon, which is chemical energy, and I mentioned that nitrogen is in proteins, and minerals, so those are your micronutrients. You add the water, the microorganisms are magically there, and then they start to cook. They heat up and utilize the carbon, and then you have water, CO2, and heat that comes off, and the water comes off, and steam. We carefully put our compost pile together, but then that's not the end of the story. We want to constantly monitor it. And so the things that are most important are to make sure that there's enough moisture. So a lot of people irrigate their pile. And some bigger facilities, they have pipes that run underneath that transfer water into the pile itself. And then air. So you guys saw the compost turner outside. So that does a couple of things. It takes the maybe more nutrient-rich materials that tend to settle out into the bottom or middle of the pile and distributes them back through equally again. It adds air and also moves that water around so that it's more evenly distributed. And that's important because, again, we have to think about what happens when a pile goes anaerobic. You get ammonia volatilization off, and that's one of the things that you're going to want to monitor. I'll show you how we can do that. And that's important because it tells you how efficient the compost pile is, but more importantly, how much of a nuisance you are to your neighbor, because that's a lot of what people smell. And then an anaerobic process is inefficient, it's slower, and so your time between the beginning So back then to the same diagram. So if there were just two things that you wanted to monitor, one would be temperature, and the other probably would be ammonia. But we can also fairly easily measure CO2 evolved as well. And so those three things are what I'm demoing here, and we would want to take a look at, especially between about the second week and probably day 60. So a lot of the bigger facilities, they monitor the CO2 and the heat and ammonia. So some other tests that you can do if you're selling compost or you're giving it to people would be this germination test to verify that there's nothing toxic in it, that's not the whole process, and that seedlings can germinate in the compost itself. So maybe you guys have seen us put together compost piles, but they're not very expensive. These thermometers for compost piles are essentially like meat thermometers in the kitchen, but they're on a long prong, long sticks, so that you can get them directly in the center of the compost pile. And so it's important to monitor it throughout the entire pile and to take measurements from the outside of the pile, partway in down to the center, so that you can verify that the majority of your pile is at that monofilic phase.
We got some information, even if you are meat-free, not to label it if you sell it.
That's a good point.
You're going to get blamed for it regardless if those seeds are in your compost or not, in what form. Your own ground or something else, your compost will be blamed for it.
Yeah, and that's why I mentioned about the incident with the pesticide too, because composting is great, and it's a shame people in many states are concerned about selling it or marketing it because of one or two incidences, and we only have to have one in a decade to have a really big impact. Go ahead, Chris.
And it's not also only the temperature, but also the duration of the heat that kills off your bacteria, your seeds and that. But also it varies from species to species. Your foxtail, that'll die out for a couple days at 130 degrees, whereas you take something like wild buckwheat, it needs about 145 days for almost a week in order to properly sterilize those seeds.
Some of the guidelines, at least for certification for organic, shifted the time frame for how long the temperature of the pile had to be for composting. People got very upset for that reason because you can't apply pesticides, and then for pathogens it's very similar. So although we want to reduce as much as we can the time that we have our compost cooking, we also don't want to compress it too far because we can take some risks if we do that. So somewhere in the range of 2 months is reasonable. Okay, so this diagram represents a chart, and this company, Salvita, that does the Salvita kit, what they do is they monitor ammonia and they monitor carbon dioxide, and then they assign a number to it, and then you find your number for the CO2 concentration and ammonia concentration, and you end up somewhere on this chart. And so based on your ammonia and CO2 together, you can kind of figure out if you have a fresh mix, which would suggest maybe that you need a little bit more carbon in there. If you were in the ideal active range, if your C to N ratio is possibly too high, and if your compost is mature. So people have been doing this without a test kit for decades already. And you guys know, about the CO2 level, maybe your temperature that you need for your pile to progress in the manner that you so desire. But this is a nice way to represent what's going on. So you could actually figure out how much CO2 is coming off the pile by taking an acid and trapping the CO2, like boric acid, which some people use as an eye wash or something, the color indicator in it. And then you can estimate the CO2 that's coming off by taking hydrochloric acid and neutralizing the CO2 that you trapped in boric acid. Oh, sorry, sodium hydroxide. Sodium would be an acid and would be titrated base. So that's what this kit is doing. It's essentially trapping CO2 and you're figuring out how much CO2 is there. And with the ammonia, you can trap that on an acid, oxalic acid. All right, let's talk about the microorganisms then. So we said that bacteria, actinomycetes, and the mesofauna are all in the pile. Mesofauna, which are things like little mites, those rotifers, they tend not to survive in high heat, so they're on the outside of the pile, or they're participants at the curing phase, or as it goes into the mesophilic phase. And so, as we mentioned earlier, actinomycetes and fungi and bacteria all produce these enzymes that break down things like cellulose and hemocellulose. Those are major components of plants that have carbon in them. And so here's some pictures of them. So the bacillus, there's a bacillus that lives in high heat that dominates the compost pile, is on the left there, your left. The actinomycetes are those stringy little things, and they're connected to particles, clay particles, but they also adhere to organic material. The trichoderma, which you probably saw today on the outside of the pile, it's that white layer. So those guys are usually on the outside. And fungi are very, they call them aerophiles. That means they need oxygen. They're loving of oxygen. So one of the reasons why you see them a lot on the outside of the pile. Aspergillus there is a type of aspergillus. People think of it as causing farmer's lung, for example. It's something that's on corn. But there is a version of aspergillus fungi that is thermophilic, and it's a major player in the compost pile. So similar but different in not a pathogen as far as I'm aware. To the left, the trichoderma, this fungi here, we find them in compost piles, and they're really neat because they're actually able to kill other pathogens. And so one of the byproducts of composting is nursery greenhouse type customers. And there are a few people in the country that have done some research about how you can add trichoderma to compost piles to essentially increase their numbers, and then take the compost that you make and use it to grow plants that are susceptible to rhizoctomy and things like that. And it's worked fairly well, actually. But when you put compost out into the field, you dilute the effect, and it doesn't work particularly well. So for greenhouse work, that's a possibility, and it's a possible value added, but it's a lot of work. All right, and then this is a compost food web. As you can see, it's pretty complicated, but basically, in blue they have the primary players. These guys are the bacteria, the fungi, and actinomycetes, which are bacteria, but have some similar properties to fungi, so we'll talk about them separately. And these guys are important players in things like vermicompost, which doesn't get heated to a high temperature. So worms, everybody's heard about composting your breakfast waste using them. So these guys would be more in your food pile as opposed to a manure pile or mortality. And those larger organisms, basically they just serve as shredders. And so the surface area is larger, so the bacteria, fungi, and the actinomycetes have access to the carbon more. Because remember, they produce all those enzymes to break up the carbon. So the physical breaking up that those organisms do is important. But in a big pile, you're turning it. So essentially, your turner is dealing much more effectively with those little guys would be doing for you. There's been a few articles that people have gone in and tried to figure out how many of each organism is in the pile at a different phase. This is one of the few articles from the 80s where somebody actually went in and looked at the bacteria at the thermophilic phase. And so at the mesophilic phase, you can see that bacteria are about double. So this is scientific, notation. So it's 10 to the 8. So that's and then in the thermophilic phase, it's more like 10,000 per gram of compost. So again, I guess when you look at the thermophilic stage, you can see that the thermophilic bacteria increased. So I guess in the end, the take-home point is that you have more bacteria in each of the phases, and that you have greater numbers of fungi in the thermophilic phase relative to bacteria. And then these are some of the organisms in the pile. So there aren't that many species. I guess that's something that we should point out in this figure. If you look at the number of species present, in a mesophilic pile there are 6 dominant bacteria and 1 in the thermophilic phase. Actinomyces are largely dominating in the thermophilic phase, and there are 14 different types of them. And fungi, the diversity is greater in the mesophilic phase, but there's still quite a bit of diversity. There's 16. So it's a pretty simple ecosystem if you think about the fact that in a gram of soil, we have never been able to quantify all the different types of organisms. And there are billions. So these are just their names. And I guess other than things like yeast that are in beer, you guys probably are not familiar with many of them. I guess the bottom line is the most important part about the organisms, the microorganisms, is their enzymes. And that's really what allows for all these things to happen. And that we end up with more complicated carbon at the end of the curing process, and those bacteria are actually creating carbon molecules that are also more recalcitrant. Then what temperature are we looking at as, okay, this pile is finished? So the center of the compost pile should be about 40 C within a few days. And within a few days of that, then we know we're at the end of the thermophilic phase. The bottom line is, I guess, at 60 C, you have a different population. That's the thermophilic phase. And then you get up at as high as 80 C. And the EPA requirement for the compost pile is 40 C or higher for 5 days. The temperature must exceed 55 C for 4 hours to get enteric pathogens. And most enteric pathogens survive less than 1 hour, but as Chris pointed out, there are certain things that form cysts, bacillus have cyst forms. Those particular types of pathogens could actually last much longer in the compost pile. So these are all based on indicator organisms that are typical, like E. coli, that don't have very long survival rates. So most compost piles spend way more than 5 days, exceeding 55 C. And as the pile cools off, we get a more complicated food web inside, and we get the introduction of those organisms we were talking about that would not survive. And so by the time we get to the curing phase, we don't have the issues with nuisance like ammonia and high CO2 rates. So we don't need to spend as much time monitoring those things, but you're going to see when you look at these little test strips that even though you can't smell any ammonia in the compost, there's still ammonia present. And so there's still some off-gas that occurs. And that's important because people and animals don't want to be exposed long-term to something like that because it has a negative impact on them. So these are things that we look at in the finished compost, and then we're going to use the test to look at. We're going to measure the CO2, and we're going to measure ammonia. So let's see. This is how you work the test. This is the test strip. And this is one of the jars. So they have a fill-to line. So I put compost in here. And then I wanted to measure my ammonia. So I have this little indicator, and it has a trap. It's probably some kind of acid, like oxalic acid. And it will absorb the ammonia, and it will draw the ammonia to this particular part. So you stick it in here. You seal it, and you wait for about 4 hours, and it just off-gases and gets caught on the little reader. Then what you can do is either take this color indicator the color here on the indicator to the indicator strip. And so this says low ammonia because basically I just opened the package and there's no ammonia on it. But over time, you get a change in color as the ammonia absorbs to the surface of the trap, and then you can read off the little color chart. Or if you have enough money and you are interested in knowing in greater detail what the ammonia loss is, you can get a little reader. And you can stick this into the reader and it will give you a concentration of ammonia off of the indicator. And so it'll tell you in parts per million. And so you can adjust it. It says low CO2, high CO2, and ammonia. So the low-tech way probably works as good as having the spectrophotometer in there. And from an agronomic perspective and from a management perspective, this color chart probably gives you a good enough reading that you can adjust your management based on that. That unit is, I think, about $1,000. So. But basically, what the number is, like the 1, 2, 3, 4, 5, and I think it's on the piece of paper you guys have, that tells you, okay, at 100 part per million, the ammonia is not detectable to the human nose. So that means you're not in violation as a nuisance in theory, and that your neighbors shouldn't be calling and complaining. At 800 part per million, it's hazard for long-term human and animal exposure. So if you're out there turning your pile, breathing in the ammonia, it's probably not very good for you. And then 2,500 parts per million, if you took that compost and you applied it, you might mess up your seedlings. You could burn your plants, or if you stuck something in that compost to grow it, it wouldn't do very well. So there's some practical application to this. It's very relevant. And then at 8,000, you have an extreme loss of nitrogen, and it's very noxious to plants. And at 25,000, which I doubt if you guys put your pile together, you would be in, you would have an unstable form. So remember the compost pile, if you don't build it right, it can actually blow up. I mean, it's rare that anything that extreme ever happens. You hear these stories about people putting a pitchfork in a compost pile and having enough gas in there to cause problems. So, and then the CO2, they have some similar information, what percent CO2 you have. So when you're at about 5% CO2, you're pretty anaerobic. So 2%, is you're getting limited. So this tells you a lot about whether or not you're going to be able to maintain the process of decomposition. And then this chart, it gives you a compost maturity index calculator. So if you take the CO2 evolved and the ammonia lost, you can figure out how close to maturity your pile is. So very high ammonia is 1 and very low is 5. So I always think of a higher number being higher, but it's the opposite on this chart. And then with the carbon dioxide, you increase your carbon dioxide as the number goes up. So you want low ammonium and low carbon dioxide. So the maturity index would be a higher value on the carbon, lower on the ammonia. I showed you guys this before. This just takes that numbering system and it gives it a description. So supposedly you'd have mature compost if your ammonia was 5, very low, and your CO2 was between 6 and 8. So in summary then, composting is a microbially driven process, so that we kind of think of as the microbes as the black box. They're a really important part of our system, and they enable us to take this very raw set of materials, break it down, and turn it into something more humic-like, similar more to soil organic matter than the starting process. And they add to that carbon themselves, stabilizing it. And at each of the stages, there's a different community of organisms that are dominant, and that would without them there, we would not be able to proceed forward with the process, and that we need to constantly monitor 3 things: temperature, oxygen, which we can do via the CO2 concentration, and then the ammonia, ideally. And so hopefully the take home here is that you can simplify the role of microbes and composting by just monitoring a few things in a very complicated system, and be very effective doing that.
The reader itself is pretty expensive. We don't have one, but these little detectors here, you can actually buy them for about 20 bucks for a pack, which would be a carbon and ammonia one. So if you really are interested and want to do that, they're about $20 a